Hi everyone, I'm Russell Ramage. I'm a food technologist with CAFRE based at our Loughry Campus. Today I'd like to talk you through some guidance that you might find beneficial if you're planning to conduct a microbiological shelf life analysis on some of your food products. This is one of two presentations on this topic, and if you'd like any more guidance or assistance, you can get in touch with us using the contact details that are at the end of this presentation. Thank you for watching. Shelf life is described as the time between the production or packaging of a food and the time when it becomes unacceptable under certain environmental conditions. Generally, unless food products are made for immediate consumption, they're required to have a shelf life attributed to them. The shelf life acts as a guide to the consumer of the period of time the food product can be kept before it starts to deteriorate. This is provided any storage conditions set by the food business operator have been followed throughout the storage, distribution, retail and consumption stages. It's the responsibility of the food business operator that the food they place on the market is fit for human consumption and free from levels of microorganisms that would otherwise present a risk to human health. A properly established product shelf life can help with an assurance to the food business operator that this is indeed the case and that they are conforming with the legal requirements of food safety that we'll look at later in this presentation. A product shelf life begins from the time a food is manufactured or prepared, and it's the time period over which a food will maintain its safety and quality. The length of a food product shelf life will depend on many factors, including the quality of the ingredients, the manufacturing process, the type of packaging used, and how the food is stored, and we'll look at some of these in more detail later in the presentation. A properly designed shelf life analysis will give you an idea of how long the product will last and its quality throughout. Careful assessment of shelf life, along with a full understanding or knowledge of the microbiological risks involved with the product, will help the food business operator avoid putting the consumer and themselves at risk. A properly conducted shelf life study will describe how the food retains its quality throughout distribution, storage, retail and final use by the consumer, and has many advantages which we'll look at later in the presentation too. The main focus of any shelf life is food safety. Many factors affect the safety of food and lead to variation in shelf life, and there's no simple answer as to how long a shelf life should be, but a properly conducted shelf life study will help food business operators accurately estimate, set and validate the shelf life of their food products. There are many reasons why it might be necessary to conduct a shelf life analysis. For instance, you could be launching a new product onto the market that needed to be date labelled. Food product innovation is a key element of the food technology work at our Loughry Campus. And if you'd like further information, you can contact us using the contact details at the end of the presentation. In the last slide we talked about ingredients and their effect on product shelf life. The intrinsic nature of your chosen ingredients can affect the length of the shelf life you assign to your food products. So for instance, if you have a product ingredient with a long shelf life because it's dry with a low water activity, and this ingredient is added to a wetter product with an increased water activity, then the shelf life of that ingredient is likely to be reduced and it should be validated as such. Also, if you change the way your products are packed, for example, that would be another occasion where a shelf life analysis should be conducted. Changing products to include modified atmosphere packaging and vacuum packaging may increase the shelf life of a food in comparison to its storage as a fresh product. These technologies can extend shelf life, since removal of oxygen is a key factor for preservation by inhibition of the growth of aerobic spoilage bacteria. This can lead, however, to an increased risk from some types of anaerobic and facultative and spoilage organisms and food poisoning bacteria, which will require additional controls for a shelf life greater than 10 days. and we'll look at these later when we consider the legislation surrounding food product shelf life aside. You might also want to reinvestigate an existing shelf life with a view to reducing food waste or in response to a request by an existing customer. The food and drink industry has a pivotal role in ensuring that consumers understand the date marks on food and drink products and use them in support of food safety and waste reduction. A number of individuals and groups have an influence on the shelf life of a product. Growers and producers have an important role to play. Quality inconsistencies or variation of the raw materials they provide can make it difficult to make a consistent final product. Raw material specifications can help maintain consistent quality and reduce these variations, leading to a more controlled shelf life. The food business operator is responsible for determining a shelf life that's suitable for their foodstuffs and one which satisfies the requirements of their food safety obligation, and also provides consistently quality products with an economically viable shelf life. Distributors have a key role to play in influencing shelf life. Any storage and food safety considerations established by the manufacturer during the design of the shelf life must be adhered to during transport and distribution. It's crucial that temperature control and package integrity are observed throughout the supply chain, and any delays in loading or unloading should be avoided, and cross-contamination is also to be avoided. Retailers and consumers also play an important role. Proper storage, handling and display of the product immediately before it reaches consumers is essential to maintain the assigned shelf life, and it's crucial that retailers do this in accordance with the manufacturer's instructions. Consumers are the final link in the chain and must be provided with accurate and correct information on storage conditions of the products. During the design of the shelf life study, it's important to remember they may not consume all the food immediately on opening, so a secondary or open shelf life must be allowed for, taking into account factors in relation to consumers' domestic kitchens and the conditions therein. There are of course many benefits to be gained from the completion of a properly conducted shelf life study. As well as satisfying the required legal obligations, food business operators can gain an assurance that the food products they are placing on the market are satisfying food safety protocols established in their HACCP systems. Also, an understanding of the growth of spoilage organisms throughout the length of the shelf life will allow the manufacturer to establish use-by dates, which give a better assurance of customer satisfaction, product longevity, and better financial management of products and associated waste management control. It's important though that the established shelf life is accurate so that food safety and spoilage concerns are met by accurate use-by dates assigned to the food product from the shelf life study. If the shelf life is too short, you will incur high manufacturing costs and reduced profit margins. If the shelf life is too long, however, that causes potential for growth of spoilage organisms and pathogens. All foods have characteristics that affect the growth of microorganisms, which in turn influences food product shelf life. Broadly split into two groups, they are the intrinsic factors and the extrinsic factors of the food. Controlling a combination of these can act as hurdles to the growth and survival of spoilage and pathogenic organisms that otherwise might shorten product shelf life. Intrinsic factors is the name given to characteristics which are inherent to the nature of the food itself, and for some of these there will be a range within which microorganisms grow best and shorten shelf life. This is because all microorganisms have optimal requirements for survival and growth. The bacterial microflora of foods and ingredients can be naturally present or from post-process contamination. And the initial contamination level and type of contaminant microorganism plays a very important role in product shelf life. For example, if your initial contamination level on fresh poultry was around 100 organisms per gram at 5 degrees centigrade, you could possibly achieve a 6-day shelf life. Raising this level to 10,000 bacteria per gram may reduce the shelf life to 4 days. pH is another important intrinsic characteristic affecting food product shelf life. It's a measure of how acid or alkaline a product is. Water has a neutral pH of 7 and it sits in the middle of the pH scale. Bacteria have an optimal pH and a range over which they grow best. A manipulation of pH can place a hurdle in the way of bacterial growth and help to prolong shelf life. For example, acidification of products can greatly extend shelf life, as is in the case of products such as cottage cheese, yogurt, fruit juices, and other naturally acidic products, and sauces which have been acidified by addition of natural acids like lemon juice. Microorganisms need water for growth. Water activity is slightly different from the moisture content of the food, and it's the amount of water in any food freely available to bacteria. For instance, pure water has a water activity of 1. Spoilage organisms and pathogens need relatively high water activities to grow and spoil products. They generally won't grow below a water activity of 0.85. So therefore reducing water activity by adding salt or sugar or using other preservation processes like drying, cooking or freezing can prolong a product shelf life. Spoilage may still occur, however, from growth of some yeasts or moulds, which can grow below a water activity of 0.6. And you should be cautious when designing a shelf life trial for some foods, since multi-component foods may have variations on water activity throughout the product. Extrinsic or external factors are the ones to which the food is subjected. Of these extrinsic factors, temperature is probably the most important one, as it greatly affects the length of time a food remains safe to eat or of a suitable quality for consumption. Inadequate temperature control of high-risk foods has been shown to be a very common cause of food poisoning and is critical to food safety. And there are normally significant increases in bacterial populations as the storage temperature of food increases. For example, in the fresh poultry case we looked at earlier, if you begin with 100 bacteria per gram at 5 degrees centigrade and increase the temperature to 10 degrees centigrade, you reduce the storage time from 6 days to 4 days. With 10,000 bacteria per gram, that changes storage time from a shelf life of 4 days to 2 days. Psychotrophic spoilage bacteria and pathogens can survive and grow at temperatures at or below 5 degrees centigrade. So chilled storage throughout a food product shelf life will considerably slow down the growth of these organisms, but won't inhibit them completely. Food business operators should set shelf life based on the careful control and monitoring of temperature throughout its design, since temperature can have such a major influence on food product longevity. And consistent application of temperature regimes to control pathogen and spoilage growth is very important to extend shelf life and control pathogen survival. You should also give consideration within reason as to what might happen after the food leaves your control, since there is likely to be a degree of temperature abuse during storage, distribution and use. This should also be planned into the shelf life, since there is a legal requirement to allow for a degree of mishandling or abuse. For example, while the pack might suggest storage at less than 5 degrees centigrade, research has demonstrated that domestic fridges regularly operate above 5 degrees. Therefore, the shelf life should account for domestic storage temperatures that are more likely to be closer to 8 degrees centigrade. This allows for the application of a buffer or a safety margin between the shelf life obtained under ideal conditions and those that are more likely to happen. For foods that are stored as fresh products, Aerobic spoilage organisms and pathogens can greatly reduce their shelf life and can potentially contribute to reduced food safety. The earlier spoilage examples we looked at were for fresh poultry in an oxygen-rich atmosphere, which led to a shelf life of between 2 and 6 days. The removal of oxygen and its replacement with other gas mixtures or a vacuum can lead to the extension of shelf life, particularly when used in conjunction with good temperature control or control of other characteristics as we discussed earlier, like pH. Reducing the concentration of oxygen in the atmosphere in which the product is stored and replacing it with a mix of gases like carbon dioxide and nitrogen can effectively inhibit the growth of aerobic pathogens and food spoilage organisms. However, many spoilage organisms and pathogenic bacteria are facultative anaerobes. This means that they can switch to different pathways used to produce energy for growth and subsequently product spoilage. Manipulation of the gas atmosphere will extend shelf life, but it won't prolong spoilage indefinitely, as the byproducts of these facultative anaerobes may eventually lead to the production of off odours and flavours. The main aim of legislation surrounding food product shelf life is to ensure that food is produced safely. This is achieved by implementation of food safety management systems based on HACCP principles, which establish safety criteria through control of foodborne pathogens. Predictive modelling and challenge testing can be used to assess pathogen growth, and we'll touch on these in more detail in the next presentation on this topic. The application of microbiological criteria through legislation gives some degree of assurance that the food is safe to eat, is of a suitable quality, and that it will remain so to the end of its shelf life. Testing against these criteria provides a measure of how well food manufacturers control the production process and control contamination, and can be used to validate if HACCP-based procedures are controlling food safety and quality. They can be used at any stage in the food chain to assess product acceptance and are based on absence or presence of certain organisms or quantitative limits of these or other organisms at any stage of a product shelf life. Anyone who packs or sells food that is required to be date labelled is legally responsible for calculating how long their products can reasonably be expected to be kept without changes to its microbiological quality. The legislation defines shelf life as the period corresponding to the period preceding the use by or the minimum durability date. In essence, the period based on identified hazards during which the product maintains its microbiological safety. Microbiological criteria have been harmonised in legislation by the European Commission regulation on the microbiological criteria for foodstuffs. That's number 2073-2005 that you see in the slide. Various interpretive documents on the legislation have been produced by organisations like the Food Standards Agency, the Chilled Food Association and the British Retail Consortium. And these are some of the main pieces of legislation that relate to shelf life testing listed here. They're easily accessible online, but just to summarise them here, they say, amongst other things, that food shouldn't be placed on the market if it's unsafe, injurious to health, or unfit for human consumption. They also say that labelling must not mislead consumers and obliges food manufacturers to recall or withdraw unsafe food from market. And they also say that the cold chain should be maintained for food that can't be stored safely at ambient temperatures. This is an example of some of the types of guidance available for food business operators considering establishing a food product shelf life. It's a decision tree allowing you to decide whether or not testing for Listeria monocytogenes should be part of your shelf life testing for ready-to-eat foods other than those that are stipulated within the guidance. In foods that have been cooked, the presence of Listeria monocytogenes can indicate undercooking or a degree of post-process contamination, and they are regarded as indicators of process or product hygiene. Listeria monocytogenes is a critical foodborne pathogen due to characteristics that allow it to grow at low temperatures and survive relatively harsh conditions such as drying and salting. It's a facultative anaerobe, which also means it can survive and grow in modified atmosphere packaging and packed foods throughout the course of a shelf life. Here you can see in the box outlined in red some intrinsic and extrinsic characteristics that you can use to establish if testing for Listeria monocytogenes should be part of your food product shelf life. If it is, then you need to demonstrate that the organism levels don't exceed 100 bacteria per gram at any point in the shelf life. With vacuum packed and foods packed in modified atmospheres, air has been removed from the packaging and either remains removed or is replaced by a mixture of gases, usually oxygen, carbon dioxide and nitrogen. These techniques can extend shelf life by inhibition of aerobic spoilage organisms, but they may lead to the growth and survival of anaerobes such as non-proteolytic Clostridium botulinum. Spores of this organism are widely distributed in the environment and may be present in food. In the right conditions, these spores can germinate and lead to the production of a very potent toxin. This can happen as low as 3 degrees centigrade. So these types of chilled foods need other controls in place to control growth and toxin production if the shelf life is to be extended beyond 10 days. It's a responsibility of the food business operator to ensure the shelf life they set is appropriate and food safety is assured throughout its length. Challenge testing and predictive microbiology may be used as a guide on how to establish and validate shelf life of these products, and this slide shows some of the controls that may be used to extend shelf life of these products. This is a possible timeline or a suggested protocol you might choose to follow if you were to decide to conduct a new food product shelf life study. There's no set timeline to follow, although often food manufacturers, in consultation with their major customers, may agree a timeline or a launch date for new or reformulated products. The design of the test regime and which microorganisms should form part of the shelf life analysis are crucial to the establishment of food safety and product shelf life, and help and guidance can be obtained on this for a number of sources, including specialised testing laboratories or from the CAFRE Food Technology team at our Loughry Campus. And finally, a few do's and don'ts to help you with the process. Do make sure that your established shelf life is accurate, real, and satisfies your legal responsibilities of food safety and quality. To help with this, make full use of the technical guidance and expertise that exists. Don't assume a one-size-fits-all approach, and remember that things can change with product reformulation or other changes that a food product may experience, including those outside the control of any food business operator. There will be another presentation on this topic in the future and available on CAFRE TV. In the meantime, if you require any further information, assistance or guidance, please get in touch with us at the contact details listed, or you can find us at Discover CAFRE on Facebook, Twitter and Instagram.